Hello students, welcome to the theory class of Principles of Plant Breeding. Today we'll discuss on breeding methods for self-pollinated crop plant, cross-pollinated crop plant and asexually propagated crop plants, which is polyploidy breeding, which can be applied to all these kinds of crop plants. Okay. So before we jump into the topic, we'll discuss some of the important terms, some of the important chemicals. Okay. So first of all, we have colchicine. What is a colchicine? It is a toxic chemical extracted from a weed species called Crocus autumnale. Okay, and its chemical nature works in such a way that the chromosome number of an individual gets double when it is treated. See, and it also works on the basis of somatic sections. Okay, so for example, if you're going to treat one of the branch with colchicines, okay, for chromosome doubling, and the another branch is untreated, so there won't be any change in the chromosome ploidy level, and in case of the treated one, there will be change in the or doubling in the chromosome level. Okay, so diploid individual will produce tetraploid individuals, and when tetraploid individual is treated with chromosome doubling with colchicine, the chromosome will be doubled and it will produce octoploid. Okay, so it can be in the form of tablets, it can be in the form of liquid, so we can purchase it for use in a lab to polyploidize our plant samples. So next we have here the chromosomal aberrations. We have already learned about chromosomal aberrations. These are changes in the chromosome at the chromosome level. So it can be either in numerical basis, it can be either in structural basis, and in case of numerical basis we have two types. Okay, one is euploids. So in case of euploids, what are these? These are change in the number of genomes. Okay, say for example 2n diploid change to triploid, triploid change to tetraploid, and so on. While in case of aneuploids, this is change or increase or decrease in one or two chromosome in a set in a diploid individuals. Okay, 2n plus 1, 2n minus 1, 2n plus 2, 2n minus 2. Okay, so today in polyploid breeding we are going to discuss broadly about euploids because they are more significant to plant breeding. And next is genome. What is a genome? It is a set of chromosome. Okay, it is a set of chromosome and it is represented by a gametic chromosome number. Okay, so in this chapter we are going to discuss the genome, and it is going to be represented by either AA, BB, CC, DD or R. Okay, so don't get confused with genes. These are not genes. We're going to represent the genome, the whole set of chromosomes as AA. Okay, homologous AA, BB, and these are not genes. Okay, and in the right side we have here is a set of chromosome, which is, you can term it as genome. So what is the chromosome number of human? 2n is equal to 46. Okay, 46, half of it is n is equal to 23. Okay, here we have 22 sets of chromosome. Okay, and plus one which is from which may be either X or which may be either Y. Okay, so if it is female, it will be both X and X, while in case of male, one of the gamete will be of Y type and another one will be of X type. Okay, so these are called allosome. Okay, and the rest of the body chromosome, somatic chromosome, are called autosomes. Okay, so this is an example of a genome, and then you can also cite the examples of rice genome. So in rice genome we have 12, n is equal to 12, so we have 12 sets of chromosome. It can also be represented, it is usually represented in circular map, and you can don't get confused here. It is all the 12 chromosomes are represented here: 1 2 3 4 5 6 7 8 9 10 11 12. Okay, so there are 12 chromosomes arranged in circle, and in this case it is arranged in vertical or horizontal vertical manner. Okay, so by the end of this class you'll be able to answer the following questions about polyploidy breeding. What is polyploidy? You'll be able to understand the type of polyploidy, what are polyploids, how are polyploids originated, and what are the morphological features of autopolyploids and allopolyploids, and with their examples. Coming to the first one, let's see what is polyploids and what is polyploidy. It involves three or more sets of chromosome. When an individual has uh, instead of 2n sets of chromosome, diploid set of chromosome, if the individual has 3 sets of chromosome, 4 sets of chromosome, or even more than that, it can be called as polyploidy. Okay, so remember, uh, chromosome set, for an individual to be called as polyploid, it should be having more than 2 sets of chromosome. That means 2 sets of haploids. Okay, and it may be either induced naturally, spontaneously, or it can be induced by artificial means. So talking about the origin of polyploids, okay, so these are the sources: somatic doubling during mitosis, okay, and non-disjunction in meiosis leading to production of unreduced gametes. And next is polyspermity, which is the fertilization of egg by 2 male nuclei or more than that, or even more than that. Okay, and endoreplication, which is replication of DNA without cytokinesis. Okay, so let's see one by one what are these. So in the first one we have here is the somatic doubling during mitosis. So this chromosome doubling happens at the level of somatic body cells, okay, and not in the reproductive cells. So that's why the non-disjunction, which is the main cause of somatic doubling, chromosome doubling, occurs in the somatic cells. Okay, so in this, what happens in the mitotic metaphase, so all the chromosomes gets aligned in the plate. Okay, and when at the anaphase, it will get separated in the poles. Okay, so proper disjunction is there when there is normal separations. Okay, and there will be and gametes in both the poles, while in case of unnormal somatic doubling, or you can say non-disjunction case, there won't be separation. Okay, there will be inability to separate between these sister chromatids. Okay, and there will be all these will go into one of the pole. Okay, so if this happens, so in all this, so the whole chromosome will move to one of the poles. Okay, so this may cause this may be a cause of somatic doubling in most of the crop plants. Okay, you can see here an example here. Diploid individuals, okay, produces diploid individual will produce male gametes 1 and 1. Okay, one is to be fused with the female gamete, which is egg cell forming zygote. Okay, and later on as an embryo, and then the one of the male gamete will fuse with polar nuclei forming an endosperm. Okay, this what happens in normal case. Okay, while in case of somatic doubling, what will happen, the 2n somatic cells will double. Okay, one maybe in one of the branch which bears the flowers. Okay, and into diploid into tetraploids. Okay, and what will happen from the pollen which is produced from this tetraploid branch, it will be diploid. Okay, both the male gametes will be having diploid. Okay, and when it is fused with a tetraploid and a tetraploid branch, okay, so it will again form what, 4n. Okay, and then and the endosperm will be 6n. Okay, so this is the main changes, okay, of somatic doubling, and the main cause is mitotic non-disjunction. Just remember this. And next is formation of unreduced gametes due to non-disjunction. Okay, so this is a second case which is a little bit different from the first case. Okay, in this case, the non-disjunction will happen in the reproductive cells. Okay, so non-reduction could due to meiotic non-disjunction, failure of the chromosome to separate, and subsequent reduction in the chromosome number. Okay, so in this case, it will be in the case of reproductive cells, which so happens in examples can be given as Raphanobrassica. Okay, so this is a synthetic what you can say allotetraploid. Okay, so likewise, the main cause is non-disjunction, which is non-separation of the non-sister chromatids in this case. Okay, and due to that, what will happen? The both these chromosomal sets will move into one of the poles. Okay, which increase in, which there will be doubled in the chromosome set in one of the gametes, and one of the gamete will have lack of chromosomes. Okay, and the next one is polyspermaty, which is the fertilization of an egg by multiple male nuclei. Okay, so it is observed in many orchid species, many ornamental crop species. Okay, and this so happens in, okay, like if you compare between the regular fertilization, the male gamete is produced in one. Okay, while in case of polyspermaty, there will be production of uh, two which fuses with the egg cell, and two which fuses with the polar nuclei, forming this, okay, triploid and tetraploid endosperm. While in other case also it can be like uh, two male gametes fusing the egg cell, forming a triploid zygote, while in case of other male gamete which is produced, it may be in just single fusing with the polar nuclei. Okay, this can be another case. Okay, so the zygote may be uh, triploid instead of diploid. Okay, the next thing is endoreplications. Okay, endoreplication is the replication of DNA without cytokinesis. Okay, although we have already learned that for with every mitotic cycle there should be, with every DNA replication there should be division of the cells. Okay, right after that. Okay, so S phase is what? S phase is the synthesis phase, that means DNA replication phase, and after that the mitosis phase works in such a way that the the double chromosome or double DNA is divided into two cells, okay, with the formation of all the proteins and RNAs during G1 and G2 phases. Okay, so if in certain cases if it escapes the cell division stage, okay, or also called as, you can call it as cytokinesis, which is what? Cytokinesis is division of the cell into two. Okay, while karyokinesis is division of the nucleus into two. Okay, so if the karyokinesis happens multiple times, and if there is no cytokinesis, there is no division of cells, there will be multiplications of huge amount of um, nucleus in a particular cell. Okay, so that is the cause of, that may be a cause of the polyploidization in a cell. Okay, so this can happen in two cases. One is endocycle, which is what? Which escapes both mitotic cycle and G1 phase. Okay, so there will not be mitosis, there won't be division. Okay, and there won't be cytokinesis. Okay, and in the endomitosis case there will be mitosis up to anaphase. Okay, so right after metaphase, at the start of anaphase, it will move to directly to S phase. Okay, so there won't be cytokinesis. Okay, and the cell will, even though the cells are divided, even though the chromosomes started to divide, move to opposite poles. Okay, but there won't be uh, there won't be two different cells. It will remain, all the chromosomes, the segregated chromosomes will remain in the same cells. The next, coming to the next slide, we have difference between haploid and monoploids. Okay, this is very important here because there is a lots of confusion. Some of the students have lots of confusions between haploid and a monoploid. Though this may represent, sometimes this may represent the same thing. Okay, in what case? In case of diploid organisms. Okay, let's see what is a haploid. A haploid of a cell is a single set of unpaired chromosomes, such as a gamete. Okay, and what is monoploid? A monoploid is having a single set of chromosome. Okay, it is denoted by N, and it is denoted by X. So if we talk about the the Mendelian cross, okay, so we have, when we cross between capital T and small t, there will be capital T, small t, okay. The gametes that is produced here are as such, okay, male gametes and female gametes. So what does it represents here? Haploid are the genetic composition of this particular gamete, one of the gamete, okay. So it will be the genetic composition of only this and not the small t, okay. So what will be that? What will be that? It will be N, okay. N will be coming from this if it is a, provided if it is a diploid, okay. And in that case female also, if it is diploid, it will be N, okay. So you all know, after division, after going through meiosis, so the 2N is reduced to N as a gamete, okay. And when we talk about monoploids, okay, it usually signifies the genetic constitutions, okay. Say for example, even though we have, in a haploid individuals, even though we have two, say for example 4 and, okay, the tetraploid individuals, okay. So the tetraploid individuals will have 2n each haploids, okay, instead of two monoploids. It won't be called as monoploids, it will be called as haploids, okay. So the tetraploid 4n will have 2n here, okay. And the 2n which is contributed by this gamete is called haploid, okay. And what is monoploid? It is the simplest genetic constitution or simplest set of the chromosome, okay, of the and what is haploid? It is the gametic number of chromosomes in a set of genome, okay. And usually in case of diploid organisms, the monoploid and haploid will be same, as you have already witnessed. For diploid, if you take diploid 2n as an example, there will be n, okay, while in the monoploids will also be x, okay. But in case of tetraploid 4n, there will be 2n, okay. But if you say the monoploids will have how many? 2x chromosomes, okay, and 2x chromosomes here. And coming to the polyploidy, okay, the different types of polyploidy, or you can say euploidy. Here the the first type is autopolyploid, which and second type we have here is allopolyploid, and the third type, which is the intermediate between the two, which is called a segmented allopolyploid. Let's see one by one. What is autopolyploid? It is the product of doubling of the same chromosome set, okay. Say for example the chromosome set is A. If it is double, it is called as diploid, the normal type, okay. And if it is triple, it is called as triploid, okay, which is represented by 3x, okay. And if it is tetraploid, it will be represented by 4 sets of the genome, okay, or the chromosome set. And if it is pentaploid, it will be represented by 5 sets of the chromosomal set, okay. And they are derived from within a single species, okay. They are derived from within a single species. That means the chromosomal set, the simplest genetic constitution, the genome, is of one type. If it is contributed from other types, which you can see here, if the genome will be of two types, okay, A and B here. So coming to the second type, we have allopolyploids. It is a product of interspecific hybridization, when you cross between two wide species, okay. So there will be obviously the genome will be different, okay. And the genome, say for example A is crossed with B, it will produce several types of allopolyploids, okay. So coming to the first type of allopolyploid we have here is AABB, okay, which is also known as amphidiploids. What are these? Two copies of two distinct genomes. Okay, 2x1, one type of genome, and 2x2, which is another type of genome. Okay, forming AABB. Okay, in case of the second one, we have two copies each of three distinct genomes. Okay, 2x1, 2x2, and 2x3. Okay, say for example AABBRR. Okay, in case of critical, and two copies of four distinct AABBRRDD. Okay, so this can also be happen in tetraploid, sorry, octaploid critical. Okay, and in case of intermediate situations, say for example if there is some of the segments is allopolyploid and some of the segments are uh, autopolyploids. Okay, say some of the segments, if you take this as an example, some of the segments here is allopolyploid. Okay, repeated in multiples of the same kind of chromosomal set. Okay, and there will be multiples of only two types of all these sets. Okay, A is represented twice. Okay, B is represented twice, D is represented twice, while R is represented four times. So there is tetraploid R, diploid D, and diploid B, and diploid A. So all these compose into the segmental allopolyploids. Okay, coming to the next slide, we have some of the morphological and cytological features of autopolyploids. These are having larger size cells than diploids. Guard cells of stomata are larger, and the number of stomata per unit area is less in polyploids than diploids. And also the pollen grains in polyploids are generally larger. This may be a consequence of the added extra genome in the polyploid species. Okay, and the growth and development is really slow in this type of and even flowering. Okay, because of the extra added genome. Okay, and polyploids usually have larger and thicker leaves and large flowers and fruits, which are usually less in number than in diploids. Okay, all the organs, okay, all the flower, all the plant parts are large, but though their number may be little bit less compared to the normal diploid species. It generally show reduced fertility due to irregularities during meiosis and due to genotypic imbalance leading to physiological disturbances. Now this may be a demerit for some of the breeding objectives when you have to go for crop plants, when you have to breed a crop for seed purpose. Okay, because there is no seed, sometimes the seed get degenerated and the pollens get infertile. Okay, pollens and male and female gametes get infertile because of the polyploidy. Okay, but in certain cases if you need seedless fruits, okay, that may be an advantage, added advantage. Okay, and in many cases autopolyploidy leads to increased vigor and vegetative growth. Okay, so overall there is a increase in growth and development. Different species have different levels of optimum ploidy. For sugar beet the optimum level is 3x. Okay, and sweet potato is 6x, while for timothy grass it is 8 to 10x. Okay, so there had to be you have to breed the crop plants based on their optimum level of ploidy. You don't have to, since they are yielding more, their fruit size is more in higher ploidy level, you don't have to go for unnecessary go for higher ploidy level, you have to go for optimum ploidy level. Okay, and autopolyploid generally have lower dry matter content than diploids. Okay, most of the autopolyploid species are have more amount of moisture content rather than their dry content. When it comes to dry content, diploids are more when compared to autopolyploids. Okay, so coming to the next slide, we have triploid, okay, which is a category of autopolyploids. So what are examples of triploids we have here? Watermelon and banana. Okay, you can see here the example of banana. The banana becomes large. Okay, there is the leaf and the seeds inside the fruit is less, which makes the fruit palatable. Okay, and instead of this, if there is huge amount of seeds inside it, people won't be uh, able to consume it, okay, fresh. Well, in case of watermelon also we have here larger fruit size and at the same time seedlessness in the fruit, okay, makes which makes the consumers easy to buy, okay, makes the market more attractive. Next thing is autotetraploid. Okay, some of the example is potato and peanut, although there are other crop plants which also possess this. Let's say the genome here represented here is PPPP, because I'm going to represent here the genes involved here as capital A and small s. Okay, so the genomes here are represented as PPPP as autotetraploid. Okay, and within these or the 4 different repeated genomes, okay, or chromosome sets, the genes present are the desired genes present are, one of them is the dominant allele, and rest of them are, if rest of them are recessive alleles, it will be called as simplex. Okay, and sometime if two of the alleles present is dominant and two are recessive, it will be called as duplex. If three of the desired alleles is three of the desired allele is dominant and one is recessive, it will be called as triplex, while quadruplex is called in those individuals where the desired gene is having all the dominant alleles. Okay, nulliplex where there is no dominant alleles. Okay, this can be classified, this can be an important question in competitive exams, so do remember this, it is frequently asked. And when say for example there is segregations, okay, since we are talking about simplex, quadruplex, duplex, what if we cross this, what if we sell this or cross between this, there can be different mechanisms. So when we do this, there will be the gametes will be having capital A small a in one and small a in one. Okay, likewise here when we have a selfing of simplex, okay, so there will be crossing between these two types, simplex types. Okay, in female gametes will produce capital A small a first as one of the gamete and small a in second gametes. Okay, likewise in the male gametes, from the male gametes it will also produce the same, so it will yield, okay, the progeny is capital A duplex, simplex and a nulliplex in the ratio 1:2:1. Okay, so do remember this, these are important for competitive exams. Okay, and coming to amphidiploid. Okay, what is amphidiploid? Presence of which is a combination of two different sets of chromosome derived from two different individuals. Okay, so when we talk about this, okay, we have two different genome sets, diploid sets from two individuals, okay, two distant individuals. Okay, so when we make a cross between this, there will be sterile hybrid. Because of the incompatibility, there will be sterile hybrid, okay, forming capital A B. Okay, and how do we make it work? By doubling the chromosome number, we can make it fertile hybrid. Okay, because in sterile hybrid we won't be able to get the seeds, the growth and development will be very degenerated. So in case of if we double the chromosome number, there will be increase in vitality, increase in vigor, okay, and then the seeds, okay, which is most important thing. And talking about amphidiploid, one of the most important example is Brassica U triangle or Nagaru U triangle. Okay, so which is the formation of amphidiploids here, AABB, BBCC, AACC with their parents. Okay, so you can see here a triangle, okay, called Nagara U triangle or Brassica U triangle. So this triangle represents in every edges of this triangle, it represents the parental genome, the parental origins, okay, parents. So these are AA, where the genome, the diploid chromosome number is 2n is equal to 20. Okay, n is equal to then 10. Okay, C is equal to and then C here, which is represented by Brassica oleracea and Brassica nigra representing BB. Okay, and BB. Okay, and in this case, so when we are crossing, when we have a cross between A B, so there will be a BB type which has a chromosome number of 2n is equal to 36. Okay, which is a combination of both. If you add these two, there will be 36. Okay, and if you add this B and C chromosome complement, there will be 2n is equal to 34, while if you add between A and C, there 2n is equal to 38. Okay, which is Brassica napus. Okay, so these are the one on the sides of the triangle are your amphidiploid. Okay, the product of interspecific hybridization, while on the corners, the edges, you have the parents. Okay, do remember this. And coming to the next one, we have evolution of natural polyploid, which is most important thing, bred with, okay, represented by AABBDD as their genetic constitutions. So first of all, their origin starts with a wild species, diploid species called Triticum monococcum, okay, which have the chromosome set AA, okay, and which also in some countries it is cultivated as Anconwheat. So it is initially crossed with a wild diploid, which is not specifically known, which is suspected to be Triticum cerci. Okay, so in this case, it represents it is the source of BB genome. Okay, so when they are crossed, there will be sterile hybrid ABB, AB, and in nature, chromosome doubling takes place, and a wild triploid tetraploid occurs, which is called Triticum turgidum. Okay, AABB, and when the Triticum turgidum is cultivated for a long period of time, which say for example 10,000 years, and as emerwheat. Okay, so when it is cultivated, so right after that, a wild species, another wild species called Triticum tauschi is crossed with it in nature and forming ABD sterile hybrid. Okay, after crossing, after doing chromosome doubling within this, okay, ABD, it forms AABBDD, which is the current bred with hexaploid or allohexaploid with Triticum aestivum. Okay, so this is the story of evolution of the bred with. Okay, coming to the next one, we have artificial allopolyploids. Okay, which is the development of artificial type of polyploids. Say for example, Raphanov brassica. First of all, in this case, radish. Okay, which is having a genomic constitution of n is equal to 9, and cabbage is crossed to produce an F1 sterile. Okay, when the F1 sterile is double using colcation treatment, the chromosome is double using colcation treatment, it will produce Raphanobbrassica, which is an amphidiploid. Okay, so likewise here same radish is crossed with brassica, cabbage, it will produce a sterile F1 hybrid. Okay, and then when it is chromosome, when the chromosome is double, the sterile F1 hybrid chromosome is double, it is fertile and it produces the amphidiploid Raphanobbrassica. Okay, so usually the motive of the aim and objective of crossing is to get the roots of the large root of radish and the leaves of cabbage, but it turned out to be the opposite of it. So there was degeneration in the root, okay, and there is prominence in the leaves, okay, which nowadays are used as Raphanobbrassica is used as commercial forage crop. Okay, and now next thing is triticale, which is also famous and because of the current use of it in some countries. Okay, and first of all, the origin is by artificial means Triticum turgidum, which is one of the wheat variety. Okay, although it is not bred wheat, it is crossed with sickle cereal, which is called rye. Okay, and when they are crossed, it produces ABR F1 sterile. When the chromosome is double, it produces AABBRR genetic constitutions allohexaploid. So when the wheat and rye, the triticale will have a totally different genetic constitution, a totally different morphological feature of the grains. Okay, so because of the luster and because of the uh, high yield in critical and quality, so they are more preferred and they are grown in some countries. Okay, and next one is this is allohexaploid. Although allohexaploid is also cultivated there, allooctaploid is also there. Okay, so this is what this is the cross using one of the parent as breadwheat instead of Triticum turgidum. Okay, if we use breadwheat, it will be AABBDD. When crossed with RR, it will produce ABDR. Okay, which is F1 sterile. If we double the chromosome, it will be AABBDD and RR. Okay, coming to the next one, we have breeding of autopolyploids. Okay, so which is the production of autopolyploids. Okay, by it may be by spontaneous, it may be a production of adventitious buds, it may be by due to treatment of physical agents, it may be during regeneration in vitro, it may be during colcation treatment or any other chemical agents. Okay, let's see one by one. In spontaneous uh, chromosome doubling, it occurs occasionally in some tissues and unreduced, and the gametes gets unreduced and are provided with and are produced in very low frequencies. Okay, although we have already seen this in the first case. And next thing is production of adventitious buds. Okay, so when you particularly pinch out the shoot tip from the plant, there will be protrusions. Okay, in the lab there will be production of callus. Okay, and from this callus there are chances that this callus will develop into the multiple plants. Okay, so the plants which is produced from this callus, okay, are sometimes leads to commonly leads to development of polyploid species. Okay, so this is a chance method. We are not going to impose any chemical, any physical agent, so we're just going to look out from the or select from the plants which is developed from the callus, from the cutout shoot tip regions. Okay, next thing is physical agents using heat or cold treatments. Okay, or centrifuge. Heat or cold treatment, as you already know, high temperature or low temperature or freezing temperature can sometimes induce polyploidization. Okay, although the frequencies may be very low. And centrifugations, in this case it is spinning of the plant samples in a centrifuge tube. Okay, this may also induce increase in number of genomes. Okay, and also X-ray and gamma ray irradiations may also produce polyploids. Okay, also although all these are chance event. Okay, we are not going to get every time we are going to do. And the next one is regeneration in vitro. What are we going to do? The polyploidization is a common feature in cells cultured in vitro. Okay, the shoot tip that we have cut out from the plants. Okay, we can grow it. Okay, so when we grow it, we can treat with some any kind of physical agents, chemical agents. Okay, like colcations, it will develop into the the polyploids. Okay, diploids. Okay, so it can occur in cell cultures of Nicotiana, Datura, rice, and several other species has been tested over it. Okay, and next thing is colcation treatment. Okay, which is most commonly used chemical and most commonly used method to double the chromosome. Okay, it is the most effective and most widely used treatment, and repeated treatments should be given at brief intervals to double the chromosome numbers in a large part of the shoot effects. So and the things so happen like this. Okay, so when you have a whole plant, if you treat the colcation in part of the plant, part of the branch, so there will be chromosome doubling in only part of that plant and there won't be chromosome doubling in other part of the plants. Okay, and last thing is application. Its application, allopolyploids application in crop improvement. First of all, as a uh, bridging of a cross. Okay, say for example, if you take uh, the hybridization of or evolution of wheat. Okay, AA is crossed with BB. So if we take this as an example, so this, okay, the genome, uh, if A is not able to cross with this, A is not able to cross with D directly, so B can be used as a bridge species. Okay, when you cross between this, B has a homologous chromosome which can pair both A and B, D genomes. Okay, so because of this, it B acts as a bridge species between A and D. Okay, although all the significant features, okay, significant characters are located in A genome and D genomes, B act as a bridge species between A and D. Okay. Next is creation of new crop species. Allopolyploids sometimes helps in creation of new species because uh, doubling chromosome number gives what? Gives a totally different type of morphological features. Sometimes it may accidentally leads to crossing of two different unknown types, and chromosome doubling may lead to totally new plants like Tritical, Raphanobrassica, and many others. Okay, and then interspecific gene transfer may also happen, and that means in case of unavailability of desired characters within the species, the gene transfer can be done with related species. And the next thing is tracing the origin of cross species. Okay, when allopolyploids are studied, okay, because of the differences in their genetic constitutions, okay, within the individual, so its origin, natural source of origin, can be tracked down from the study. Okay, so coming to the end of the slide, so if you have any questions you can ask.